Revolutionizing Biology. Scientists create E. coli with 57 codon genetic code. The creation of SYN57 marks a groundbreaking advancement in the understanding of life's genetic framework. This innovative synthesis of E. coli, utilizing only 57 codons, represents a significant overhaul of the genetic code, demonstrating that life can thrive with 7 fewer codons than previously thought. For billions of years, the genetic code has remained nearly universal among all known life forms, employing 64 combinations of DNA and RNA bases. Referred to as codons, to encode 20 amino acids and stop signals essential for protein synthesis. However, this system is not without its flaws. Many codons redundantly instruct for the same amino acids or stop signals. Jason Chin's research team at the LMB's PNAC division previously achieved a remarkable feat by designing and synthesizing an entire E. coli genome with just 61 codons, known as SYN61. This accomplishment illustrated that life can indeed operate with a more streamlined genetic code. Now, the team has taken a bold step further by eliminating 7 codons to create SYN57, a strain of E. coli that operates with only 57 codons. In a remarkable effort led by Wes Robertson, Fabian Reim, Martin Spink, Raphael Schumann, and Rongzhen Tian, the researchers removed 4 of the 6 codons responsible for encoding the amino acid serine, 2 of the 4 codons for alanine, and 1 stop codon. These were replaced with synonymous codon substitutes that can produce the same end products. This ambitious project involved recoding over 101,000 codons within a synthetic genome of 4 megabases. To tackle this extensive task, the genome was segmented into 38 fragments, each approximately 100 kilobases in size. These fragments were synthesized individually using homologous recombination in yeast, before being inserted into E. coli through a method called uREXER. This technique is a variant of the replicon excision-enhanced recombination (REXER) method, which combines CRISPR-Cas9 technology with lambda-red recombination to replace genomic DNA with synthetic DNA in a single step. During this process, the team identified challenging regions of the genome that resisted recoding or exhibited reduced growth rates. They employed various strategies to address these issues, such as modifying the N-terminal coding sequences to enhance gene expression, restructuring overlapping genes to facilitate independent recoding, and fine-tuning the codon substitutions. After completing each fragment, the team executed iterative uREXER steps to assemble them into a series of semi-synthetic strains. Throughout this process, they utilized a bacterial mating technique to converge the semi-synthetic strains into a final, fully synthetic organism. The outcome, SYN57, stands as the most extensively recoded organism to date, showcasing that life can indeed function with a significantly compressed genetic code. Jason's team has previously demonstrated that their SYN61 strain can be reprogrammed to incorporate non-canonical amino acids, enabling the synthesis of entirely new polymers. By eliminating 4 additional codons, SYN57 offers even more potential for introducing non-canonical amino acids, paving the way for the development of innovative synthetic polymers and macrocycles. Furthermore, similar to SYN61, future research on SYN57 will explore its inability to interpret the canonical genetic code, which could render it resistant to viral infections. This advancement has the potential to reduce costs and enhance reliability in drug manufacturing by mitigating the risks posed by viruses. This groundbreaking research was supported by various funding bodies, including UKRI MRC, the European Research Council ERC, the Wellcome Trust, the German Research Foundation DFG, UKRI Marie Skłodowska-Curie Actions, the Boehringer Ingelheim Fonds, the Cambridge Commonwealth, European and International Trust, and the Harding Distinguished Postgraduate Scholars Program. For further reading, you can explore the following references. Escherichia coli with a 57 codon genetic code. https://www.science.org/doypine/ADY4368 by Robertson et al. Jason's group page, https://mark.mp.cam.ac.uk/group/leaders/A to G/Wes Robertson. Additionally, previous insights on related research can be found in articles such as Refactoring the Genetic Code to Create Organisms Protected by a Genetic Firewall, https://refactoring the genetic code to create organisms protected by a genetic firewall. Cells reprogrammed for genetically encoded polymer synthesis and viral resistance, https://mrc.mp.ac.uk/cells reprogrammed for genetically encoded polymer synthesis and viral resistance. Scientists redesign E. coli genome with 101,000 changes, a breakthrough in synthetic biology. In a groundbreaking scientific feat, researchers have achieved the unprecedented task of redesigning the E. coli genome with over 100,000 modifications to its DNA, pushing the boundaries of synthetic biology to new heights. This monumental effort, led by a team at the Medical Research Council Laboratory of Molecular Biology in Cambridge, UK, has resulted in a bacterium that utilizes just 57 of the 64 genetic codes, a significant reduction that could have far-reaching implications for the future of biotechnology. The creation of SYN57, the 57 codon E. coli, represents a significant leap forward in our ability to manipulate life at the genetic level. The team's primary objective was to demonstrate the feasibility of such a feat, but the potential commercial applications of SYN57 are equally intriguing. With further genetic tweaking, this bacterium could become completely resistant to viral infections, a game-changer for the industrial production of proteins used in medicines, food, and cosmetics. This is because viruses require their host's genetic code to synthesize proteins, and by altering the code, viral proteins would be rendered dysfunctional. Moreover, SYN57 could potentially produce proteins containing up to 27 different amino acids, a significant increase from the 20 found in natural proteins. These synthetic proteins could possess unique properties and functionalities that are unattainable with conventional proteins, opening up new avenues for innovation in various industries. A protein is essentially a chain of amino acids assembled according to the sequence dictated by a gene. Each triplet of DNA letters, or codon, instructs the protein-making machinery on which amino acid to add next, or when to halt production. With 64 possible codons and only 20 amino acids used by living organisms, there is considerable redundancy in the genetic code. By replacing all instances of one codon for a specific amino acid with another codon for the same amino acid, the original codon becomes available for alternative uses, such as coding for a non-natural amino acid or a different chemical, enabling the creation of novel proteins. In theory, up to 43 codons could be repurposed in a living organism, as only 21 are necessary. 20 for the standard amino acids and 1 for the stop signal. However, this is currently unattainable due to the increased likelihood of detrimental effects with more extensive genetic modifications. Instead, biologists are starting small, with 314 gene edits made to E. coli in 2011 to free up one codon. To expedite the process of making thousands of gene edits, Wesley Robertson and his team opted for synthesizing DNA from scratch. In 2019, they unveiled SYN61, a version of E. coli with 18,000 changes to its 4 million DNA letters, freeing up 3 codons. Constructive Bio, a spin-off company, is now exploring the commercial potential of this achievement. The latest iteration, SYN57, involved 101,000 changes to free up 7 codons. This required testing small fragments of the recoded genome in living bacteria to identify and correct any detrimental effects. The process was repeated with increasingly larger fragments until the entire genome was complete, a testament to the team's perseverance and scientific prowess. Akos Nirgis, a researcher at Harvard Medical School, acknowledges the significance of this achievement, which is the result of years of dedicated work. Nirgis's team is also working on freeing up 7 codons in E. coli, but by recoding different codons. Their 57 codon E. coli strain is still in development. While SYN57 is already a complete organism, it grows much more slowly than its natural counterpart. For commercial viability, this growth rate will need to be improved. Robertson is optimistic about this prospect, stating that they anticipate being able to enhance the growth rate, making SYN57 more useful for industrial applications. For the time being, Robertson's team intends to concentrate on exploring the potential applications of SYN57, rather than attempting to further compress the genetic code. There is much work to be done before considering additional genetic code compression. The first bacterium with a synthetic genome was created in 2010, but the goal was to create a simplified organism rather than recoding codons. This latest achievement, however, marks a significant milestone in the field of synthetic biology, pushing the boundaries of what is possible and paving the way for future advancements in biotechnology.